はい、それでは定刻が近づいてまいりましたので、まずちょっとご紹介から、させていただきたいと思います。皆さんどうもおはようございます。朝早くからお越しいただきまして誠にありがとうございます。またオンラインの参加者の皆様もどうもありがとうございます。私、このセッションの座長を務めさせていただく京都大学の白髪と申します。こちらのセッションは、厳密に言うと、アクアフォトミクス学会の、正式なプログラムの方からはちょっと外れていまして、スポンサーセッションという扱いになります。今回は、このスポンサーセッションの中で5件の、発表いただくことになっておりまして、時間が10分、15分の、発表が、2件、3件それぞれあるというような形になっております。このスポンサーセッションなんですけれども、基本的に学会、本体の方では、一切の撮影と、禁止しておりますけれども、こちらのスポンサーセッションに関しては、ご了承をいただいておりますので、自由に写真撮影と動画撮影、していただいて構いません。ということで、よろしくお願いいたします。じゃあ、それでは早速1件目の方から入りたいと思いますので、ご準備の方よろしくお願いいたします。The first presenter is Yuda-san from Aquaphotomics,
Yes.
Yunosato Aquaphotomics Lab, and his presentation title is on the pilot study exploring solid dynamics using near-infrared spectroscopy and aquaphotomics. So if you are ready, please start.
Okay, thank you very much. Good morning to everybody. Good morning to everybody. My name is Yuda Hadiwijaya. I'm from Indonesia, and now I'm a researcher at Yuno Little Aquaphotomics Laboratory. Today, I would like to present some of our experiment results about the applying aquaphotomics in the agricultural area. Specifically, it is exploring soil dynamics using near-infrared spectroscopy and aquaphotomics. Soil is essential for the plant growth because soil provides nutrients for the plants. Soil can retain the water. Soil also helps microbes to make the nutrient available for the plants. That's why soil monitoring is important to ensure the optimal condition of the soil. However, generally, researchers and scientists measure the properties of soil using an instrument or a method that can only measure one parameter. Of course, it is costly and time-consuming. In the other hand, aquaphotomics is a new approach that study the water-soil relationship. It offers the cost-effective method because it can measure several parameters in one measurement. It's also fast and non-invasive, which means we can just take the soil surface spectra and leave the environment undisturbed. So the objective of the experiment was to explore the role of water in soil characterization, soil water retention properties, monitoring of plant development, and we also wanted to investigate the effect of organic fertilizer on soil and plant. For the near-infrared spectroscopy measurement, we did at our laboratory, Yuno Little Aquaphotomics Laboratory, Wakayama, and we use two types of instrument. This is about our having a similar specification. This is our first experiment. We collected three types of soil: agricultural soil, garden soil, and forest soil. We did air drying after that, and then we continue to measure the spectra of the samples. This is the aquagrams of the first overtone for comparison between the agricultural, garden, and forest soil. You can observe that the agricultural soils contains the higher water sulfation shell, trap, and free water molecules, while the forest soils is in the opposite direction of the agricultural soil. the first direction with highest organic matter content generating the most hydrogen bonding of water molecule. And this is our second experiment. We did rewetting on each of types of soil to get the moisture content level start from 0, 1, 5, 10, 20, and 50% of moisture content level. The bottom pictures are the raw spectra of the first overtone, start from 1300 to 1600 for each types of soil. Then we can observe that the higher moisture content leads to higher absorbance level. So the difference between agricultural and garden and the forest soil, for the agricultural soil, we can see the very well separation between each moisture content level, which means the agricultural soils is the most sensitive to the changes of moisture content level. This is our exploratory analysis using PCA scores. Here we did PCA on each moisture content level regardless the soil types. There are two groups, drier and the wetter soil. So and also we create the aquagram for this moisture content level. And for the drier soil, it is rich in a vapor-like water molecular conformation, while the wetter soil, with the increased number of small and big water cluster. In the next experiment, this is the third experiment, we tried to grow the carrot using the natural way. We didn't apply any perturbation, we didn't apply any fertilizer. We just wanted to see how the soil spectra changes during the crop development. So we did monitoring for the spectra during the seedling stage, vegetative, and the harvest stage. And then the aquagram results, there is a complementary spectral pattern relationship between soil and leaves. For the leaves aquagram at the vegetative stage, we can see it contains higher free water molecules during the vegetative stage. The free water molecule is very important for the plants during the vegetative because it supports the growth and metabolism. The plants during vegetative stage are performing a lot of photosynthesis, while in the harvesting stage, the photosynthesis is going down. So the leaves contain more hydrogen bonding because they reduce metabolic reaction and increase the preservation properties. This is our fourth experiment. In this experiment, we wanted to investigate the influence of organic fertilizer to the soil properties. And then we have three groups of treatments. The vegetables with soil, only with soil without any fertilizer. And then the second treatment is planted soil plus solid organic fertilizer, which is the pig manure. And in the third treatment, we use the planted soil plus solid and liquid organic fertilizer. We assume that by applying the liquid fertilizer, the plants can absorb the fertilizer easily compared to the solid one. And as you can see, from the growth of the plants, there's an enhancement. By visually, we can see it by visually and also by the spectral pattern. So the organic fertilizer optimize the microbial growth. It increases the water solvation shell, trap water, free water molecule, hydronium ions, and water dimmer. And also the organic fertilizer, it stabilizes the availability nutrients, the essential nutrient for the plants. like nitrogen, phosphate, potassium, and the calcium. And then for the conclusion, the aquaphotomics successfully applied for exploring the soil dynamics non-invasively, characterization for soil types with prominent water absorption band, distinct properties activate through varying soil hydration states, spectral correlation, there's spectral correlation between soil and plants, and the plant and microbial growth stabilization achieved through the time with specific cultivation technique. Thank you very much.
Okay, thank you for your presentation. 冒頭にお伝えし忘れていましたけれども、こちらのスポンサーセッションは、すべてちょっと質疑応答は、なしとさせていただきます。と言いますのも、今回スポンサーセッションで話される皆さんは、皆さんはポスターでも、発表されることになっていますので、時間の関係で、いろいろ議論されたい方は、後ほどのポスターセッションで深く議論いただければと思います。